The cell membrane. Introduction to the cell membrane. What is cell membrane? Cell membrane is a dynamic, flexible boundary that separates the internal contents of a cell from its internal and external environment. Usually, in the plant cell, the cell membrane is the second outermost, while in animal cell, it is the outermost cell. It is also referred to as the plasma membrane in some contexts. Now, there is a model which explains it very well, which is known as the fluid mosaic model. And fluid mosaic model says that it describes the cell membrane as a fluid, dynamic structure where lipids and proteins can move laterally, giving it a mosaic-like appearance. Okay, so that's why it is known as fluid mosaic model. Importance: it acts as a gatekeeper. It allows something, it does not allow something. It's also known as the selective permeable membrane, and its adaptability is crucial for growth, reproduction, and interaction with the environment. Historical milestones. Overton hypothesizes a lipid nature of the membrane. So he said about the lipid membrane of the membrane. Then Goddard and Grindel proposed the lipid bilayer theory, which was accepted before. And the most recent in 1932 is the fluid mosaic model introduced by Singer and Nicholson, which is the most authentic one. Detailed points. Lipids provide the structural basis while proteins execute most membrane function. The surface of the membrane is unique to the cell type, contributing to cell identity. As you can see here, there is a carrier protein. This is known as the carrier protein, and these are the channel protein, and there is phospholipids, which is made up of phospholipids, and then there is a glycoprotein. We got cholesterol and integral protein as well. Structure and function. The first and important thing in cell membrane is the phospholipid bilayer. The fundamental structure of the membrane is a double layer of phospholipids. It has a hydrophilic head and a hydrophobic head. A hydrophilic means water attracting, water loving, and the hydrophobic tail means the water hating, like water repelling. And then we got proteins. In protein we have integral protein and we have peripheral proteins. Integral proteins span the entire membrane and are involved in transport, cell transduction, and enzyme activity, while the peripheral protein are attached to the surface of the membrane and are involved in structural support and signaling. Then we got the cholesterol. Not in that much quantity, but still there is. It is embedded within the bilayer. Cholesterol molecules stabilize the molecule membrane and maintains its fluidity, especially in varying temperature. And the last but not the least is the carbohydrates, often attached to proteins, glycoproteins or glycolipids on the extracellular side of the membrane. It plays a role in recognition, signaling, and adhesion. Functions of the cell membrane. What are the functions of the cell membrane? Cell membrane is really important in the cell. The first function is selective permeability. As I told you in the previous slide, that the membrane regulates the entry and exit of substances. It is also known as selective permeability membrane. It usually allows the smaller molecules like the water molecule. It does not allow the big molecules. Then we got transport. It facilitates the movement of substances through passive transport and active transport. Passive transport is basically diffusion and osmosis, no energy required, while for active transport there is requirement of energy, ATP. Cell communication. Through cell membrane, cell can communicate. Cell adhesion. Two cells can do adhesion, like they can stick together with the cell, because of the cell membrane structural support. It provides a flexible yet sturdy boundary that maintains cell shape and protects the internal environment. Now, there is difference between integral proteins and peripheral proteins in the location. So, integral protein are embedded in the membrane, while the peripheral are attached to the membrane surface. And function. The integral have function and channel receptors, while the other one have support communication. Difficult to remove the integral one, while the other one is easy to remove. Relatively insoluble, while the other one, the peripheral, is soluble. Transport across the cell membrane. There are two types in which the things can be transported across the cell membrane. One is passive transport, and the other one is active transport. For now, we will talk about passive transport. What is passive transport? Passive transport is basically, it does not require energy and relies on the natural environment. Molecules down their concentration gradient, means they will go from high concentration to low concentration. The first and most important type is simple diffusion. Movement of small non-molecular molecule, for example, oxygen, carbon dioxide, directly through the phospholipid bilayer. For example, oxygen entering cells for respiration. Diffusion is basically net movement of molecules from high concentration to low concentration through the concentration gradient. While facilitated diffusion, it is exactly the same thing as simple diffusion. There's only small difference, which is facilitated mean there will be like specific transport routine which will help in the diffusion process. So it is providing like a facility. The third topic is osmosis. Osmosis is also a type of passive transport. Osmosis and diffusion are relatively same. There's only a small difference. It is basically also known as diffusion of water. Water molecule through a selectively permeable membrane. Water moves from area of low solute concentration to high solute concentration. For example, if there is low solute inside the cell, the water will go inside. If there is high solute concentration, low solute concentration outside, the water will go there. Now the second type which we have to study here is active transport. So as I told you before, in active transport there is usually ATP required. ATP is required to move substances against a concentration gradient. Now this is also another difference, that in active transport the substances will go from low to high concentrations. This is the main difference as well. Types of active transport. The first one is primary active transport. The second one is secondary active transport. It is also known as co-transport. What is the difference? So it is direct use of ATP to power the movement of molecule, while the other one is uses the energy from an existing gradient created by primary active transport to move other substances. Exocytosis. Exocytosis are vesicle fused with the plasma membrane to release substances outside the cell, for example, secretion of hormones or neurotransmitters. These all topics are discussed in the next slide. We will see like in the picture that how this happens. The fourth one is specialized transport mechanism like ion channel, like electrochemical gradient, and the third one is coupled transport. What is ion channel? Allows selective movement of ions, for example, sodium ion, potassium, and chlorine, based on charge and size, can be gated, open, or closed in response to signals. Then we got electrochemical gradient. The combined effect of concentration gradient and membrane potential influences ion movement. And the last but not the least, coupled transport. Substances like glucose are transported alongside ions like sodium ion with specific supporters. Transport types. Now this is just like a summary. In simple diffusion, energy is not required in facilitation. There is no only in secondary active transport there is energy required. In exo there is energy required. In primary there is energy required. From high to low, high to low, high to low, low to high, low to high, and in or out bulk. So these are all the examples, direction and energy required. This is exocytosis. So what happens is, for example, as you can see here, there's like solutes from inside which is needed outside, which is needed inside. So it comes here and it forms like a vesicle, and then the vesicle is engulfed by the cell. Cellular structure and their specific function. Cells contain various structure called as organelles, each one with specific function essential for cellular activity. We will study each organelle one by one. The first one is plasma membrane, then we got nucleus, then we got the mitochondria. These are the three most important organelles. Plasma membrane or the cell membrane, phospholipid bilayer with embedded proteins. It acts as a selective barrier, regulating the entry and exit of substances. It facilitates cell communication and adhesion. Structure. It is enclosed by a double membrane nuclear envelope with pores, contains nucleoplasm, chromatin, and nucleolus. Its function is houses genetic material DNA that controls cellular activities. Nucleolus synthesizes ribosomal RNA and assembles ribosomes. The third one is mitochondria. It is a double membrane organelle with an inner membrane folded into cristae. Its main function is it produces ATP through cellular respiration. It is also known as the powerhouse of the cell because most of the energy of the cell is produced in the mitochondria. Then we got ribosomes and endoplasmic reticulum. And ribosome is the place where the protein is synthesized. It is composed of RNA and proteins found free in the cytoplasm or attached to the rough endoplasmic reticulum. So it is either free in the cytoplasm or attached to rough endoplasmic reticulum. It synthesizes protein by transferring messenger RNA. Then endoplasmic reticulum, it is a network of membranous tubules and sacs. There are two types. First one is smooth endoplasmic reticulum. It is stuffed with ribosomes, while the other one is more smooth endoplasmic reticulum, which lacks ribosomes. Which doesn't have. So the rough one have ribosome, while the smooth one does not have ribosomes. Its function: the rough endoplasmic reticulum synthesizes and processes proteins, while the smooth endoplasmic reticulum synthesizes lipids, detoxifies chemicals, and stores calcium. Golgi apparatus. We got Golgi apparatus number 6, peroxisomes, and we got vacuoles. Now, what is Golgi apparatus? It is a staked, flattened, membranous sac. Its function is to modify, sort, and packages proteins and lipids for transport or secretion. Peroxisomes. It is a membrane-bound organism containing oxidative enzymes. Its function is to break down fatty acids and detoxify harmful substances, for example, hydrogen peroxide. It breaks down the hydrogen peroxide vacuole. It is membrane bound. It takes larger in plants. Its function is to store nutrients, waste products, and water. In plants, the central vacuole maintains turgor pressure. So this is how plant cell look like. Plant cell is usually like a rectangular shape, while animal cell is rounded. In plant cell, there is like a big vacuole in the middle, and there's chloroplast, which is not available in animal cell. There is a cell wall, which is not available in animal cell, and here in animal cell, it has lysosome, which is not in plant cell, and yeah, that's it. Membrane dynamics and cell communication. The plasma membrane plays a central role in regulating interactions between the cell and its environment, its dynamic nature, and communication mechanisms. We will study these two one by one. Membrane dynamics, the first topic. The plasma membrane is a fluid mosaic structure allowing constant movement and reorganization of its components. This fluidity is essential for various cellular processes, including transport signaling and membrane repair. The first one is lateral movement. Phospholipids and proteins move laterally within the bilayer. It enables flexibility and the redistribution of membranes. The second one is selective permeability. The membrane allows specific molecule. Remember, it allows specific molecules. Specific means it will allow the small non-polar molecule and pass freely, while the larger or charged molecule require transport proteins. The third one is membrane remodeling. The membrane can expand, contract, or reorganize during processes like endocytosis, exocytosis, and cell division. Membrane protein. There is integral and peripheral proteins in the membrane which move within the bilayer, facilitating transport, signal transduction, and interactions within the cytoskeleton. Lipid rafts. Microdomains rich in cholesterol and sphingolipids provide platforms for protein clustering, enhancing signal transduction and trafficking. Cell communication. Now the second big topic is the cell communication, which is the cell communication is, let me highlight it down. This is the main topic which we need to study. Alright, so the cell communication involves the exchange of information between the cells or between cells and their environment. It relies on signal molecules. It relies on receptors and downstream pathways. Examples of cell communication. So the first one is immune system. Antigen presenting cells interact with T cell to trigger immune responses. Neurotransmission. Neurons release neurotransmitter like dopamine to communicate with other neurons or muscles. The third one is hormonal signaling. Insulin regulates glucose uptake by binding to receptor on target cells. Membrane fluidity versus temperature. So it shows that with the high the temperature, the high the membrane fluidity will be. Specialized membrane adaptation in different cell, or it is also known as the specialized organelles. So like these are the things which are like specialized, which are not in all cells. The first and main thing is microvilli, finger-like projection of the plasma membrane supported by the cytoskeleton. It increases surface area for absorption, found in intestinal epithelial cells and in the small intestine. It mainly is involved in the absorption, enhanced nutrient absorption. Remember, the microvilli does the nutrient absorption because of the high surface area. Cilia. Now cilia is a hair-like extension of the plasma membrane containing microtubules. You can see here it is mainly in the respiratory system. It moves fluid, mucus, or particles over the cell surface. It is found in the respiratory tract. It moves mucus and trapped particles. For example, if there's like a pathogen or mucus which went to your lungs, so it will move it to outside to your mouth. And fallopian tube cells transport eggs. It is also found in the fallopian tube cells in the females. Then we got flagella. Flagella is really important for movement. Long whip-like structure made of microtubules. It propel cells through liquid environment found in sperm cells. As you can see here, this is known as the flagella. This is like tail-like structure. It is mainly used for motility, and it is also found in some prokaryotes. Tight junction. Now, there is some tight junctions between inside the cell, like specialized protein complexes that seal neighboring cells together. Function is it prevents leakage of substance between cells. So because of these tight junctions, there is a very less chance of leakage. It is found in epithelial cells lining the intestines and blood-brain barrier. The fifth one is specialized transport proteins. Specialized transport proteins are membrane proteins adapted for specific molecules. For example, its function is to facilitate selective transport. For example, glucose transporters and red blood cell ion channels in neurons. It is found in all cell types and with specific transport needs. The topic number 6 is the myelin sheet. Where is myelin sheet found? It is layer of plasma membrane wrapped around axons. It is mainly found in the axon in the neuron cell. Its function is to insulate neuron fibers and speed up electrical signal transmission, because neuron has to communicate with another neuron, so it does that function. It is found in Schwann cells, peripheral nervous system, and oligodendrocytes, the central nervous system. So it is basically found in peripheral and central nervous system. Cell wall associated membrane modification. How is cell wall associated with modification? Plasma membrane associated with rigid cell wall. Its function is support and regulate osmotic pressure. It is found in plant cell, which is made up of cellulose, and in fungi, which is made up of chitin, in bacteriophage, which is made up of peptidoglycan.